Welcome to the deep dive. You brought us a stack of sources detailing the domain Archaea,
Yeah.
and we are going deep into what these incredible microbes are, how they survive, and why they're kind of rewriting the story of life on Earth.
Yeah, absolutely. When Archaea were first, studied seriously, the perception was they were purely extremophiles.
Right, the weirdos.
Exactly. The biological oddities you only found in, like, boiling acid springs, deep sea vents, super salty lakes.
Places nothing else could live.
Pretty much. They were seen as fascinating, sure, but kind of niche, a sideline.
And what our sources really hammer home is that this view is just completely outdated now. Archaea are, well, they're everywhere.
Totally. Ecologically diverse, central players in global systems. I mean,
Right.
the numbers are staggering. Something like 20% of the prokaryotic biomass in marine plankton alone is Archaea.
Wow.
They're in soil, they're absolutely in our gut, they're not fringe dwellers at all, they're woven right into the biosphere's fabric.
And that ubiquity, that presence everywhere, is why they suddenly become so relevant to, well, current events, right?
Yeah,
Especially energy and climate.
definitely. When we talk about natural gas methane, we are fundamentally talking about Archaea.
How so?
Well, methane can form non-biologically, abiotically deep in the earth, but basically all biologically produced methane comes from one specific group of organisms, the methanogenic archaea,
Only that.
exclusively. They generate it by reducing carbon dioxide with hydrogen, or sometimes by munching on simple stuff like acetate.
And this immediately connects them to the whole debate around energy, specifically hydraulic fracturing, or fracking.
Exactly. Fracking is how we get at those huge stores of methane trapped deep underground in shale. Massive economic implications, obviously.
Huge environmental questions, too.
Right. Concerns about potentially contaminating groundwater, and also the big one, methane leakage. These fugitive emissions are a really potent greenhouse gas, much more so than CO2 over shorter timescales.
So these ancient microbes are basically sitting at the heart of a massive 21st century debate about energy and climate.
They really are. They're the quiet biological engine driving the whole thing, both making the gas originally and, as we'll see, sometimes consuming it too.
Okay, so let's unpack this. Our mission today is to explore how these fascinating microbes are actually built, how they eat their metabolism, and why their family tree, their taxonomy, is still, well, kind of a work in progress.
Sounds good.
Let's kick things off with their unique identity. Genetically, they seem to be a real puzzle.
Oh, absolutely. They're an evolutionary mosaic. It really challenges that neat three-domain tree of life we often see.
How does that work?
Well, get this. Archaea have information genes, the ones for copying DNA, making RNA, building proteins that look a lot like eukaryotic genes, like ours.
Okay.
But then their metabolism genes, the ones for, eating, breathing, building cell parts, they generally look much more like bacterial genes.
So sophisticated information processing paired with efficient microbial survival tricks.
That's a great way to put it. It's like they cherry-picked the best bits from both worlds.
And that uniqueness extends to their structure too, right? They don't just look like typical bacteria.
Not at all. Morphology-wise, they go way beyond simple rods and spheres. You see weird lobed shapes, flat squares, triangles, cells that are just irregular, pleomorphic, and sizes vary hugely, too.
But the really striking thing is the membrane, especially in the heat lovers.
Ah, yes, the hyperthermophiles. They use these special lipids called Archaeal. They're C40 isoprenoid tetraethers, big, long molecules.
And they don't form a bilayer.
No, that's the key. Instead of the two layers we see in bacteria and eukaryotes, these long lipids span the entire membrane, forming a single, tough monolayer.
A monolayer membrane, that sounds fundamentally different. How does that help?
It's all about heat stability. A monolayer is just inherently more rigid and resistant to falling apart at extreme temperatures compared to a fluid bilayer.
Yeah.
It locks everything together. It's a direct adaptation to that intense heat pressure.
Makes sense. Now, classifying these guys sounds tricky, partly because so many are hard to grow in the lab.
Exactly. Most Archaea we know about haven't been cultured. We know them primarily from sequencing DNA directly from environmental samples. That's metagenomics. So their taxonomy is heavily based on gene sequences, not lab observations.
So Berkey's manual, the sort of Bible of prokaryote classification, how does it handle them now?
It's evolving. Currently, it recognizes three main established phyla. There's Euryarchaeota, that's the really diverse one, with methanogens, extreme salt lovers, halophiles, sulfate reducers, all sorts.
Okay.
Then there's Crenarchaeota, which are mostly known as the thermophiles and hyperthermophiles, and relatively recently separated out as Thaumarchaeota, which are actually widespread, often found in moderate environments.
So sequencing keeps revealing more complexity, right? These superphyla.
Yeah, the picture's getting bigger. We now talk about the tax superphylum that bundles together Thaumarchaeota, Crenarchaeota, and a couple of others, and the depan superphylum, which seems to be mostly these really small symbiotic or parasitic archaea with tiny genomes.
And then there's the really exciting one, Asgard.
Yes. The proposed Asgard Archaea superphylum, still uncultured, known only from genomes reconstructed from environmental DNA. But the reason they're causing such a stir is that their genomes contain genes for what we previously thought were eukaryotic signature proteins.
Like what?
Things involved in the cytoskeleton, like actin and tubulin homologs. Proteins involved in complex membrane remodeling, stuff we thought was unique to eukaryotes.
So what does that imply?
Well, it strongly suggests that eukaryotes didn't just evolve alongside Archaea, but might have actually emerged from within an Archaeal lineage. Perhaps the Asgard group, or something like it, represents our closest prokaryotic relatives. It could fundamentally change our understanding of where complex life came from.
Wow. Okay, shifting from who they are to what they do, let's talk metabolism. Many of the autotrophs, the ones fixing carbon, live without much oxygen.
That's right. Most are anaerobic or live in very low oxygen environments, and this often puts them right up against their thermodynamic limits. They're basically scraping by, allergy-wise.
So they need to be super efficient.
Extremely efficient. And one key adaptation is their choice of electron carrier for carbon fixation. Instead of using NADPH, which is common elsewhere, many archaea use reduced ferredoxin, F to butt.
And why is ferredoxin better in those low-energy situations?
Simply put, reduced ferredoxin carries electrons at a much lower redox potential. It's more energetic. About negative 400 millivolts compared to NADPH's 320.
Okay.
That extra push from the electrons means they can drive reactions more easily, potentially using less ATP overall to fix each molecule of CO2. When you're living on the energetic edge, every little bit of ATP saved counts.
Efficiency's everything. And our sources lay out 3 main pathways they use to fix CO2.
Yep, 3 main routes identified. First, there's the reductive acetyl-CoA pathway, also called the Wood-Joule pathway. This one is incredibly energy efficient.
The cheapest option.
By far. It costs only about 1 ATP per pyruvate formed. And for methanogens, it's even more central because they use parts of this pathway not just for building cells, but also for the actual energy-generating process of making methane. It does double duty.
Okay, super efficient. Then there's one that's apparently the opposite. The 3-hydroxypropionate-4-hydroxybutyrate cycle, or HPHB cycle. This one's really costly.
Massively costly in terms of ATP, maybe around 9 ATP per pyruvate. It seems counterintuitive after talking about efficiency, right?
Yeah, why use such an expensive pathway?
The key advantage is oxygen tolerance. Unlike the other pathways, the HPHB cycle can operate perfectly well in the presence of oxygen, so it allows organisms like some Crenarchaeota and all the Thaumarchaeota to fix carbon aerobically.
Ah, so it's a trade-off. High energy cost, but opens up aerobic environments.
Exactly. And the third one is the decarboxylate 4-hydroxybutyrate DCHB cycle. This is another anaerobic pathway, moderately expensive, about 5 ATP per pyruvate, and it interestingly uses some steps that look like the Krebs cycle, the TCA cycle, run in reverse.
Fascinating diversity there. What about breaking down sugars like glucose? Do they use the standard pathways we see in bacteria and eukaryotes?
Nope, they do it differently there too. They often like the key enzymes for the standard pathways like glycolysis, Embden-Meyerhof, or the standard Entner-Doudoroff pathway. They've evolved novel variations.
Like the hyperthermophiles using a modified Embden-Meyerhof pathway.
Right. A key feature there is they often use kinases that depend on ADP, not ATP, and the surprising result compared to standard glycolysis.
Let me guess, less ATP.
No net ATP produced in those initial sugar-splitting steps. It's not about making energy right away. It seems geared towards quickly breaking down the sugar into building blocks, using electron carriers like ferredoxin or NAD plus muro, and setting the stage for later energy generation. It's prep work.
Okay. And similar modifications exist for the Entner-Doudoroff ED pathway.
Yeah, we see variations there too. Extreme halophiles use a semi-phosphorylative version that does net 1 ATP, But then some organisms, like Sulfolobus, use a non-phosphorylative version.
In non-phosphorylative means.
Again, zero net ATP produced during that phase. It completely bypasses the main ATP-generating step you see in the bacterial ED pathway. It's all about generating pyruvate and other precursors efficiently, not immediate energy gain from the sugar itself.
So they break things down into this key 2-carbon unit, acetyl-CoA. But cells need 3-carbon units like pyruvate as central building blocks. How do they make that conversion?
Good question. They need a way to stitch two acetyl-CoA molecules together, essentially. The most common way, used by many autotrophs and some Haloarchaea, is the familiar glyoxylate cycle.
Okay, that's standard enough.
But then, here's another really elegant adaptation. Some other Haloarchaea, like Halophrex vulcania, use a much more complicated route called the methylaspartate pathway.
Why use a more complex pathway if the glyoxylate cycle works?
Because the methylaspartate pathway has a very useful side product in their specific environment, glutamate.
Glutamate, an amino acid. Why is that important for them?
Because for a Haloarchaeon living in incredibly high salt, glutamate acts as a potent osmoprotectant. It helps balance the osmotic pressure inside the cell.
So the pathway doesn't just make building blocks, it makes something that helps them survive the salt.
Exactly. And the hypothesis is that the buildup of intracellular glutamate might even act as a signal, telling the cell about the osmotic stress and activating this specific pathway. It beautifully links their metabolism directly to their environmental challenge. Just really clever biology.
That adaptability really shines in the phylum Euryarchaeota. We mentioned the methanogens globally important.
Absolutely central to the carbon cycle. Methanogenesis is the final step in breaking down organic matter in places without oxygen. It's how they make energy, taking carbon in its most oxidized form, CO2, and reducing it all the way down to its most reduced form, methane, CH4.
That's a huge chemical transformation.
It is, and it requires this whole toolkit of unique cofactors molecules you basically only find in methanogens. Things with names like methanofuran, tetrahydromethanopterin, coenzyme M, coenzyme B, coenzyme F430. They're all involved in shuffling these single carbons and electrons around.
And the scale is immense, right? 1 billion tons of methane a year.
Roughly, yeah. Globally significant. It's vital for biogas production in anaerobic digesters, which we use for energy. But it's also, a major greenhouse gas source.
Yeah.
The classic example is a cow. Its gut is basically a methanogen bioreactor. They can belch out hundreds of liters of methane per day.
Wow. But nature has a counterbalance, this ANME process.
Yes, anaerobic methanotrophy, or ANME. It's crucial. If all the methane produced in places like deep marine sediments escaped, our climate would be very different. Estimates are that something like 90% of that deep sea methane gets oxidized before it reaches the water column or atmosphere.
And the culprits are other archaea.
Primarily, yes. Groups called ANME1, ANME2, AMA3. We still can't really culture them reliably, but genomic studies suggest something amazing. They seem to be running the methanogenesis pathway essentially in reverse. They use methane as their fuel, as their electron donor.
Running it backwards? How?
They often live in close consortia, tight partnerships with sulfate-reducing bacteria. The Archaea oxidize the methane and somehow pass the electrons to the bacteria, which use them to reduce sulfate.
How do they pass electrons?
That's a hot area of research. Some evidence, including microscopy, suggests they might form direct connections using conductive protein filaments, basically, biological nanowires, to shuttle electrons between the cells. It's fascinating stuff.
Absolutely wild. Okay, switching gears within the Euryarchaeota. The Haloarchaea, salt lovers.
Extreme salt lovers. We're talking organisms in the order Halobacteriales that require at least 1.5 molar in ACl, that's about 9% salt just to grow. Below that, their cell walls can actually disintegrate.
So how do they cope with living in brine, essentially?
They use what's called the salt-in strategy. Instead of pumping salt out, they actively accumulate salts, mainly potassium chloride, KCl, but also sodium chloride, NaCl, inside their cytoplasm to match the concentration outside.
So the inside of the cell is just as salty as the outside.
Pretty much, which means all their internal components, especially proteins, have to be adapted to function in that incredibly high salt environment.
How do proteins adapt to that?
They tend to have a lot of negatively charged amino acids like aspartate and glutamate on their surfaces. These attract positive ions like potassium and help form a hydration shell around the protein, keeping it soluble and functional even when surrounded by molar concentrations of salt. It's a major evolutionary overhaul of their entire proteome.
Incredible. And one of them, Halobacterium selenarum, has this amazing trick for harvesting light without chlorophyll.
Yes. This is one of the classic discoveries in archaeobiology. They use a protein called archaerodopsin. It contains retinol, the same light-absorbing molecule found in the rhodopsin in our eyes.
So it's like an eye protein.
Structurally very similar, which is amazing, but its function is different. It's not for vision. It's a light-driven proton pump.
It pumps protons using light energy.
Exactly. When light hits archaerodopsin, it undergoes a conformational change and physically pumps a proton from inside the cell to the outside. This directly generates a proton gradient, a difference in pH and charge across the membrane.
And that gradient is used to make ATP.
Precisely. The protons flow back into the cell through an ATP synthase enzyme, driving ATP production. It's chemiosmosis, just like in respiration or photosynthesis, but the initial gradient is generated directly by light hitting this single protein. No electron transport chain needed for this part.
Just a simple, elegant, solar-powered proton pump. Do they have other rhodopsins too?
They do. They often have Haloarchaea, which is another light-driven pump, but this one pumps chloride ions into the cell. This helps maintain that high internal salt concentration. And they also have sensory rhodopsins. These act like photoreceptors, detecting light intensity and color and signaling to the cell's motility system, the archaea, which are kind of like their flagella, to move the cell toward optimal light conditions for the pumping rhodopsins and away from potentially damaging UV light.
A whole suite of light-interacting proteins. Amazing. Let's move finally to the taxonomy, particularly the Crenarchaeota. These are the real heat fanatics, right?
Yeah, many Crenarchaeota are hyperthermophiles, often found in volcanic environments like sulfurous hot springs, solfataras, or deep-sea hydrothermal vents. Optimal growth, often above 85 degrees Celsius.
And some can tolerate even higher.
Oh, yeah. The record holder mentioned in the sources is Pyrolobus fumari. It can apparently survive being autoclaved, heated to 121 degrees Celsius under pressure for an hour. Just incredible heat stability.
What do they eat in these environments?
Sulfur is often key. Many are sulfur-dependent, using elemental sulfur, S0, either as an electron acceptor in anaerobic respiration or sometimes as an electron donor for chemosynthesis. Sulfolobus is a famous example. It's a thermoacidophile.
Meaning hot and acidic.
Exactly. Grows best around 80 degrees C and pH 2 or 3. But here's the thing. Even though the outside is incredibly acidic, it has to maintain its internal cytoplasm near neutral pH, maybe around 6.5.
How does it manage that huge difference?
It relies on a very impermeable membrane and actively pumps protons out to maintain that massive pH gradient. That gradient is then used to drive ATP synthesis and other processes.
And in these extreme places, we find some extreme relationships, like that tiny parasite.
Right, Nanoarchaeum equitans. It's currently the only cultured member of that DPAN superphylum we mentioned. It's an obligate parasite living on the surface of another Crenarchaeota, Ignococcus hospitalis.
But its genome is tiny.
Ridiculously small, under 500,000 base pairs. It seems to have lost the genes for making most essential molecules: lipids, amino acids, nucleotides. It's completely dependent on its host, Ignococcus, for almost everything. Its genome reduction taken to an extreme.
Very stripped-down existence. Then there are the Thaumarchaeota.
Not extremophiles, necessarily.
Not typically, no. These were recognized relatively recently, partly because scientists kept finding this unique lipid signature in moderate environments, like marine plankton and soils. The lipid is Thaumarchaeol.
What's special about it?
It contains a 6-membered carbon ring, a cyclohexene ring, integrated into one of the lipid tails. This structural feature is thought to help keep the membrane fluid at the lower temperatures where these organisms live, compared to the C40 tetraethers of hyperthermophiles.
And what's their main job ecologically?
They are major players in the nitrogen cycle as aerobic ammonia oxidizing archaea, or AOA. They convert ammonia to nitrite, the first step in nitrification. They're abundant in oceans and soils, often outnumbering ammonia oxidizing bacteria. And they can be flexible eaters. Mixotrophs.
Yes. Many seem capable of mixotrophy. They get energy from oxidizing ammonia, but they can also incorporate dissolved organic carbon compounds for building their cells rather than fixing all their carbon from CO2. It gives them flexibility.
And besides these main groups, there are still others known only from genes.
Oh, absolutely. Phyla like Euryarchaeota, Crenarchaeota, Bathyarchaeota.
Right.
We only know them from sequences pulled out of environmental samples. It highlights just how much archaeal diversity is still dark matter. We know the genes exist, but we haven't seen or grown the organisms. And new roles keep emerging. Archaeota, for instance, were recently inferred to also be capable of methanogenesis, a job previously thought restricted to Euryarchaeota.
The map is definitely still being drawn.
Constantly.
Okay, so wrapping this deep dive up,
Uh-huh.
what's the big picture for our listeners? What are the absolute key takeaways about Archaea?
Well, I think the first thing is they're not just extremophiles. They are a distinct, ancient, and incredibly diverse domain of life found everywhere.
Right, and defined by that unique genetic mix, eukaryotic-like info processing, bacterial-like metabolism.
Exactly. Second, they play absolutely critical roles in global biogeochemical cycles, like methane production and consumption, nitrogen cycling through ammonia oxidation. They're not minor players, they're fundamental.
And third, they achieve all this through some truly remarkable biochemical adaptations.
Absolutely. From monolayer membranes that defy extreme heat, to light-driven proton pumps like archaerhodopsin, to those incredibly diverse and sometimes highly efficient or strategically inefficient ways of fixing carbon dioxide, their biochemistry is just full of surprises.
So connecting this to the future, what's the lingering question or the next frontier?
For me, it's the sheer volume of archaeal diversity we only know from genomes. These uncultured groups, especially things like the Asgard Archaea with their eukaryotic-like proteins,
Yeah.
they raise profound questions. How much fundamental biology, how many unique metabolic tricks are hidden within organisms we can't yet grow?
We're potentially missing huge parts of the story.
We almost certainly are. Understanding these uncultured lineages, and particularly unraveling that deep evolutionary connection between Archaea and the origin of eukaryotes, that remains one of the biggest, most exciting frontiers in all of biology, I think. We've learned so much, but the Archaea story is clearly far from over.
A perfect place to leave it. Thank you for taking this deep dive with us today.